This protocol observes and analyzes cell rolling trajectories on asymmetric receptor patterns to facilitate engineering of receptor-patterned substrates for label-free cell separation and analysis. Begin by microcontact printing of PEG molecules on a gold substrate to create alternating patterns of self-assembled monolayers, followed by backfilling the substrate with p-selectin receptors. Next, incorporate the substrate with the flow chamber. Flow the HL60 cells into the chamber. Record an image sequence of the rolling interaction of HL60 cells on p-selectin patterns and analyze the images converted from the video. The trajectories of rolling cell edge tracking length rolling velocities on plane region and edge can be obtained using a MATLAB program.
This method can provide insight into how HL60 cells move on the asymmetric receptor patterns in shear flow. This can be applied for quantifying other cell surface interactions, direct analysis of the cells by observing their trajectories on receptor patterns, and for design of microfluidic devices for cell separation.
Microcontact printing is used to create alternating patterns of self-assembled monolayers of PEG molecules on the gold substrate. First, ink the PDMS stamp with 5 millimolar PEG solution in ethanol. Dry the surface for 20 minutes. Gently put the stamp on the gold surface for 40 seconds and make sure there is a good contact between the gold surface and the stamp. No excess pressure is required. Rinse the surface with ethanol and dry under a stream of nitrogen. Now using